Welcome back, class. Today's lecture is lecture 7, Control of Microorganisms by Physical and Chemical Agents. So in this lecture we'll be focusing on the control of microbes, but keep in mind that there are lots of mechanisms used to control microbes that are not covered in this lecture. So for example, the manipulation of environmental conditions is used extensively in the food industry to preserve foods. Increased solutes such as sugar and salt are used to preserve meats and jams, for example. Microbial fermentations of milk and vegetables decrease the pH of these foods, and this creates new foods such as yogurt and cheese and so on, and all of these have a longer shelf life than the milk and vegetables from which they're made. Heat and the generation of anoxic conditions are also important in preserving canned foods. And ionising radiation is used to extend the shelf life of seafood, fruits and vegetables. This flowchart very nicely summarises the major microbial control methods that are used. And you'll see that the methods are placed into one of three broad classes at the top of the flowchart, either physical agents, chemical agents, or mechanical removal methods. So physical agents in pink can use either heat or radiation, and the heat that's used can be dry or moist. Chemical agents in blue includes gases or liquids, and mechanical removal methods in green incorporates the use of filtration, that is filtration of air or liquids. Now what I'd like you to note here is the final aim of each of these control methods, and the aim is either sterilisation, disinfection, antisepsis, or chemotherapy. Chemotherapy we won't be talking about in this particular lecture, and we'll look at those terms in a bit more detail on the next slides. So terminology is actually quite important when control of microorganisms is discussed, because words like disinfectant, antiseptic and sterilisation are often used quite loosely and often incorrectly. And the situation's even more confusing when a particular treatment can either inhibit growth of the microorganisms or kill the microorganisms depending on the treatment and on the environmental conditions. So the ability to control microbial populations on inanimate objects like surgical instruments, for example, is of quite practical importance. Sometimes it's necessary to eliminate all microorganisms from an object, whereas at other times only partial destruction of the microbial population may be required. So let's look at the first term here, sterilisation, which comes from a Latin word meaning unable to produce offspring. This is the process by which all living cells, spores and acellular entities such as viruses are either destroyed or removed from an object or habitat. So a sterile object is completely free of viable microorganisms, spores and other infectious agents. When sterilisation is achieved by a chemical agent, the chemical is called a sterilant. In contrast, disinfection is the killing, inhibition or removal of microorganisms that may cause disease. The primary goal of disinfection is to destroy potential pathogens, but disinfection also substantially reduces the total microbial population. Disinfectants are usually chemical agents and they're used to carry out disinfection, and they're normally only used on inanimate objects. Keep in mind, a disinfectant does not necessarily sterilise an object because viable spores and a few microorganisms may remain. Sanitisation is closely related to disinfection. In sanitisation, the microbial population is reduced to levels that are considered safe by public health standards. The inanimate objects are usually cleaned as well by being partially disinfected. For example, sanitisers are used to clean eating utensils in restaurants. Now, it's also frequently necessary to control microorganisms on or in living tissue with chemical agents. The term antisepsis, which comes from the Greek meaning against putrefaction, is the prevention of infection or sepsis and is accomplished with antiseptics. Antiseptics are chemical agents applied to tissue to prevent infection by killing or inhibiting pathogen growth. And they're also used to reduce the total microbial population. Now, because they must not destroy too much of the host tissue, antiseptics are generally not as toxic as disinfectants. Different suffixes can be used to denote the type of antimicrobial agent. So substances that kill organisms often have the suffix "cide" from the Latin to kill in their name. So cidal agents will kill the microorganisms. On the other hand, static agents will inhibit growth but not necessarily kill the microorganisms. So the suffix "cide" indicates that the antimicrobial agent will kill the microorganisms. A germicide kills pathogens and also many of the non-pathogens, but not necessarily endospores. A disinfectant or an antiseptic can be quite effective against a specific group of microorganisms, and in this case it may be called a bactericide, fungicide, algicide, or viricide. The suffix "static" indicates that the antimicrobial agent inhibits growth but doesn't kill the microorganisms. So if these agents are removed, then microbial growth will resume. So this suffix "static" is from the Greek meaning causing to stand or stop. And some examples here: bacteriostatic and fungistatic. Now, it should be noted that although these agents have been described in terms of their effects on pathogens, they also kill or inhibit the growth of non-pathogens as well. So their ability to reduce total microbial populations, not just to affect pathogen levels, is actually quite important in many situations. So when we expose a microbial population to some antimicrobial agent, the population isn't killed instantly. Population death, just like population growth, is generally exponential or logarithmic. That is, the population will be reduced by the same fraction at constant intervals. If the logarithm of the population number remaining is plotted against the time of exposure to the agent, then a straight line plot will result. When the population's been greatly reduced, the rate of killing may actually slow down, and this is due to the survival of perhaps a more resistant strain of the microorganism. So to study the effectiveness of an antimicrobial agent, we need to be able to decide when the microorganism is dead. Not an easy task. A bacterium is often defined as being dead if it does not grow and reproduce when inoculated into culture medium that would normally support its growth. In the same way, an inactive virus would not be able to infect a suitable host. But this definition has serious flaws. It's been demonstrated, for example, that when bacteria are exposed to certain conditions, they might remain alive but are temporarily unable to reproduce. And when they're in this state, they're referred to as viable but non-culturable, VBNC. We've covered this in a previous lecture. So in conventional tests to demonstrate killing by an antimicrobial agent, VBNC bacteria would be thought to be dead. And this is a serious problem because after a period of recovery, the bacteria may regain their ability to reproduce and cause infection. To understand the pattern of microbial death, let's follow this theoretical microbial heat killing experiment. Now we're going to assume that the initial sample contains 10 to the 6 vegetative microorganisms per mil, and that 90% of the microorganisms are killed during each minute of exposure to a temperature of 121 degrees Celsius. So at the end of the first minute, 90% of the microorganisms are killed, and this leaves us with 10 to the 5 microorganisms remaining. At the end of the second minute, 90% of those microbes are killed, which leaves us with 10 to the 4 microbes remaining. At the end of the third minute, 10 to the 3 remaining. Fourth minute, 10 to the 2 remaining, and so on. And you can see that by the time we get to the end of the of 7 minutes, then we have effectively killed all the microorganisms in that sample. The same data is shown here as an exponential plot of the survivors, which you can see on the y-axis, versus the minutes of exposure to heating at 121 degrees Celsius. So the minutes of exposure you can see on the x-axis. In this example, you can see a value called the D value, D121, and the value of the D value here is 1 minute. We'll come back to what the D value means in just a moment. Now, destruction of microorganisms and inhibition of microbial growth are not straightforward matters because the efficiency of an antimicrobial agent is affected by at least 6 different factors. Let's look at the first two here. Population size. Because an equal fraction of a microbial population is killed during each interval, a larger population will require a longer time to die than a smaller one. And we saw an example of that in the theoretical heat killing experiment. The second factor is population composition. So the effectiveness of any antimicrobial agent will vary with the nature of the organisms being treated because microorganisms differ in their susceptibility to any antimicrobial agent. Bacterial endospores, for example, are much more resistant to most agents than are vegetative cells. And younger cells are usually more readily destroyed than mature organisms. Some species can even withstand adverse conditions better than others. So, for example, Mycobacterium tuberculosis, which causes the disease tuberculosis, is much more resistant to antimicrobial agents than most other bacteria. Often, but not always, the more concentrated a chemical agent or intense a physical agent is, then the more rapidly microbes are destroyed. However, the effectiveness of these agents usually is not directly related to concentration or intensity. Over a short range, a small increase in concentration leads to an exponential rise in effectiveness, but beyond a certain point, then increases may not increase the killing rate very much at all. And sometimes an agent is more effective at lower concentrations. So, for example, in the laboratory, we generally use 70% ethanol or 80% ethanol. And this is more effective than, for example, 95 or 100% ethanol because its activity is enhanced by the presence of water. An increase in the temperature at which a chemical acts often enhances its activity. Frequently, a lower concentration of disinfectant or sterilising agent can be used at a higher temperature. The final condition is the local environment. So the population to be controlled is not usually isolated, but surrounded by certain environmental factors that may either offer protection or aid in its destruction. For example, because heat kills more readily at an acidic pH, acidic foods and beverages such as fruits and tomatoes are easier to pasteurise than foods with higher pHs like milk. A second important environmental factor is organic matter, which can protect microorganisms against heating and chemical disinfectants. A good example is the biofilm. The organic matter found in a biofilm protects the biofilm's microorganisms, and the biofilm and its microbes often are hard to remove. In addition to this, it's been found that bacteria in biofilms are altered physiologically, and this makes them less susceptible to many antimicrobial agents.